Thank you very much. Firstly, I'd like to express my sincere thanks to the organizers for inviting me to this attractive symposium. So I've been working on the pathogenesis and the morphogenesis of infection structure of the anthrax-known fungi, Colletotrichum species, which include more than 100 species and infect important crops, like here, as shown here. Especially, I've been working on C. orbiculare, which infect cucurbitaceous plant as a model fungus. This fungus formed a melanized epithelial and infection structure on the leaf surface and makes hemibiotrophic infection to its host plant and expects the lesion like this. So far, the pathogenesis and the morphogenesis related factors were reviewed in Journal of General Plant Pathology as the 100th anniversary volume, collaborating with Yoshitaka Takano of Kyoto University. Those factors include melanin biosensing genes and also peroxisome functional genes and signal transduction pathways such as cyclic MP-dependent pathway and 3 MAP kinase pathway that are involved in conidial germination, apelation formation, apelation penetration, and invasive growth, respectively. And very recently, we have found that the development of epithelial, the infection structure, is under strict cell cycle control. The conserved GTPase activating protein, a two-component GTPase activating protein, GAP, which consists of BAV2 and BFA1, that plays a role in mitotic exit and Saccharomyces cerevisiae, plays a role in G1S progression and pathogenesis, in Colletotrichum orbiculare. This slide shows the mitotic stage of wild type during apelation formation. At 4 hours of incubation, the mitosis occur at the time of the apelation differentiation. However, in the GAP mutant, the mitosis occur around 2 hours of incubation, 2 hour earlier than the wild type in pre-germinating conidia. And these mutants show the defectiveness of pathogenesis and also defective in penetration hyphae formation, quite different from the wild type. So the cell cycle control is important for expressing the proper pathogenesis. So in this talk, I'd like to show our recent findings on C. orbiculare plant interactions. The talk will include 2 parts as shown in the abstract. One is plant signal recognition, namely how the fungus recognize plant and start the development of epithelium. The second part is the control or hemibiotrophy, which consists of initial biotrophic phase that's following the necrotrophic stage, and in which shift is important for evaluating plant defense responses. So let's move on. The first part, plant signal perception and morphogenesis. In our previous study, we have identified the COK2 gene of C. orbiculare. This gene is a homologue of fusion yeast, Cytosaccharomyces pombe, TAA1, that plays a role of cellular polarized growth. Accordingly, the COK2 of this fungus localized at the apical growing region, similar to the Cytosaccharomyces pombe, and involved in the development and morphogenesis. And on the glass slide, at the model substrate, the KL2 mutant formed abnormal episoria on the glass slide, quite different from the wild type. But however, interestingly, or rather surprisingly, the mutant formed normal episoria and penetration hyphae similar to the wild type and retained fruit pathogenesis as the wild type. So it was indicated COK2 was required for proper morphogenesis of episoria on condition of perceiving in vitro physical signals from substrate. So it was hypothesized that there could be a redundant pathway. One is a physical signal where in which COK2 is involved. The other is a plant-specific signals leading to the episode formation. The research target we set was to elucidate the plant signaling pathway. To achieve this goal, we conducted the forward mutant screening of forming abnormal episoria on the plant with the COK2 mutant background, namely the W mutant, considering the redundancy. And successfully, we obtained 6 mutants with expected phenotype with abnormal episoria formation on glass slide and also on the host plant, and showed reduced pathogenesis, but formed normal episoria by reintroduction of COK2 complementation, the redundant pathway. And from one of 6 mutants, named PDF2, we were able to identify the disrupted gene as Saccharomyces cerevisiae PAG1 gene. The Saccharomyces PAG1 is a component of RAM, regulation of AS2P and morphogenesis that plays a role in multiple cellular processes, including the cell separation, polarized messenger RNA localization, secretion, or stress signaling. And the most important component of RAM is a kinase CBK1. And the PAG1 is critical for RAM function as a scaffolding protein. Because AS2P is not present in C. orbiculare as like in other filamentous fungi, MOR, morphogenesis related NDR kinase network, is used in place of RAM. To elucidate the function of PAG1, we generated a gene disrupting mutant of PAG1. On the plant, the COK2 mutant formed melanized, matured normal episoria and penetration hyphae, similar to the wild type. But introduction of the COPAG1 mutation to COK2W mutant showed abnormal phenotype and the penetration hyphae formation deficiency. However, the single mutant, the episorial formation and penetration hyphae formation was normal, indicating that the redundant pathway could be functional. Accordingly, the pathogenesis of the double mutant was attenuated, reduced, while the single mutant retained full pathogenesis. So next we checked whether the PAG1 functioned through activation of CBK1, like in Saccharomyces cerevisiae. If the PAG1 functions through activation of CBK1, reintroduction of CBK1 constitutively active form will restore normal appressor formation. So as expectedly, the normal appressor formation was restored when the COCBK1 constitutive active form was introduced into the double mutant vectory. And furthermore, the CBK1 CA partially restored the pathogenicity of the double mutant. So these results indicate there is actually the redundant pathway. One is the abiotic physical pathway where COK2 involved. The other is MO pathway where the PAG1 activates COCBK1, leading to the proper appressor formation. And next question, what is the plant signal for inducing the appressor formation? So we evaluated the effect of a plant surface QT monomers, 1,16-hexadecanediol, and octadecanol. This was actually identified from the leaf surface exudate of the cucumber leaves. In the presence of these QT monomers, the normal appressor formation of COK2 was effectively restored, while the double mutant was not restored, indicating that these QT monomers function as a signal molecule. By the way, the rust fungi and the powder mildew, the spore surface esterase is involved in the attachment of host surface or host infection. So we checked whether the spore surface esterase is involved in the QT monomer production. Firstly, we checked the surface esterase activity by using the indoxyl acetate as a substrate. So we were able to detect the esterase activity on the unwashed spore, but not washed spore. In this figure, the spore is moved from the attachment site. Below the attachment site, the crystal was observed. This is the autoclaved and minor substrate contour. Furthermore, we were able to detect the extracellular esterase activity from the washed water exudate. This is the big liver contour. So next we check whether the production of octadecanol is dependent on the spore surface esterase activity. And the amount of the octadecanol was evaluated in the absence of conidia of leaf surface exudate or in the presence of spore. The result was in the presence of conidia spore, about 2 microgram ml of octadecanol were detected that were sufficient for inducing appressor development, but not in the absence of spore. So this means indicate that octadecanol is released from the activity of spore surface esterase that induce appressor formation. So the summary is that plant derives signal Qt monomers from plant surface derived from the spore surface esterase activity, function as a signal molecule, and through a MOR pathway, appreciable morphogenesis is induced. And the next question is, what is the downstream target of MOR? To achieve this goal, we selected the plant inducible genes that were affected by PAG1 mutation and CBK1 inhibition by microarray analysis. And successfully, we were able to select 10 candidate transcription factors named MTF1 to 10, and successfully obtained 8 gene disruptive mutant on the wild type background and also Kyoto background. And out of these 8 disruptive mutants, MTF4 shows the expected phenotype that with the retained pathogenesis in the wild type background that show the defectiveness, pathogenicity defectiveness with the Kyoto double mutant. Expectedly, the MTF4 Kyoto mutant formed the abnormal episoria on the host plant, but not a single mutant MTF4 Kyoto formed the normal episoria as expected. So single mutation does not affect the pathogenesis or morphogenesis, similar to the PAG1 mutation. So if the MTF4 function downstream or MOR overexpression constitutively active expressing overexpressor introduction to the PAG1 Q2 would restore normal appressor formation. And the result is that expected. The introduction MTA4 overexpressor to the double mutant restored normal appressor formation and also pathogenesis. And furthermore, the dependency of MTF4 on the MOR was further evaluated by cytological analysis of MTF4 localization. In the wild type, MTF4 GFP localized in the conidial nuclei. So the co-localized with histone, RFP, in the nuclei of the conidia, as you can see on the plant, but not on the in vitro glass slide. But the localization of MTF4 was diminished in PAG1 delta mutation or CBK1 inhibition condition. So all of these data indicate that MTF4 could be a downstream target of MOR pathway. So I'd like to summarize the conclusion of this part. Qt monomer derived from cuticle by spore surface esterase activity function as plant surface signal. The signal is transduced through MOR pathway for inducing upstream differentiation in C. orbiculare. And conclusively, C. orbiculare has organized system for the recognition of plant that lead to the development of infection structure. So now I'd like to move on to the second part, hemibiotrophy and pathogenesis. In a previous study, RAS GTPase activating protein COIL1 regulate the activity of RAS1 and 2 as an upstream regulator of cycling pathway, PK pathway, and a MAP kinase pathway. And the RAS2 dynamically localized during upstream development. And in Saccharomyces cerevisiae, a phosphatase complex we do is involved in actin dynamics and also actin dynamics as an upstream regulator. So we have an interest in the V2 function in C. orbiculare. And we generated, find the V2 homolog of C. orbiculare and generate the gene-disrupting mutant. The gene-disrupting mutant showed weaker lesion when compared to the wild type, and this is the complement one. And the most typical phenotype of this mutant is that this mutant forms very thin, narrow hyphae when compared to the wild type. The narrow hyphae is typical for the necrotic hyphae. So I will hypothesize that the first biotrophic phase might be skipped in this mutant. So next, we evaluate the defense responses of C or with inoculation. This is the data of dove staining. As you can see, the loss accumulation was much increased in weight inoculation. And also, Karos deposition by V2 under the upstream formation. This signal showed the Karos is much increased in the V2 inoculation. And also the real-time PCR analysis of the Karos synthase gene identified in cucumber. 3 out of 12 Karos synthase genes were upregulated during the V2 infection. And furthermore, the necrotrophy specific responsive defense in PDF 1.2 was much increased in the V2 inoculation. However, biotrophic stage PR1 is rather downregulated. The white box is a PAMPS mutant, SSD1, the PAMPS mutant that induces PTI used as a control. Furthermore, the biotrophy stage was evaluated by fluorescence visualization of Venus fused with biotrophic specific expression genes and an infection hyphae. The fluorescence of polysaccharide deacetylase COPSD1 promoter fused Venus was clearly detected, definitely detected in the biotrophic hyphae, and similarly, that of 4-aminobutyrate aminotransferase fused Venus was clearly detected in biotrophic phase. So using this fluorescence, we were able to evaluate the biotrophic phase. And expectedly, the Venus signal and V2 was significantly reduced in the PSD1 promoter and also ATF1 promoter. So these results, all these results indicate biotrophy is, establishment of biotrophy is under V2 control and the V2 function. So the next question is what genes are controlled by V2 function? To achieve this question, to answer this question, we conducted DNA microarray analysis in the early infection stage. This data shows the heat map of the top 150 genes upregulated in V2 inoculation. Out of them, the interesting thing is that those genes include ribosomal protein, related protein, as listed in this table. And it was reported in the mammal and Saccharomyces cerevisiae. The expression of ribosomal protein is regulated by tau signal, target of rapamycin signaling. So we hypothesized that the enhanced expression of those ribosomal genes is due to the activation of Toll signaling. And the hypothesis was supported by the application of rapamycin. So those enhanced expression was downregulated in rapamycin treatment. And furthermore, the representative two genes, RT-PCR analysis of representative two genes indicate that increased enhanced expression of V2 was downregulated rapamycin treatment. So the Toll could be underweighted Toll is functional for its functions. Lastly, we evaluated whether biotrophic establishment of biotrophy is under Toll regulation. And the data here, the calor synthase, calor deposition of the V2 inoculation increased after the inoculation, but application of rapamycin decreased the calor deposition. And also, the intensity of biotrophy-specific promoter fused venous was increased by rapamycin treatment, indicating Toll signaling is regulation. Toll signaling is important for the establishment of biotrophy. So this is the conclusion of this part. And V2 functions as a key signaling component that control hemibiotrophy through negative regulation of Toll signaling. Lastly, I'd like to show the student who concerned in this experiment. The first part of my talk, the plant recognition and morphogenesis was done by Sayu Kodama. And the second part, the hemibiotrophic regulation, is done by Ken Harata. And I'd like to thank the collaborators and funding association. Thank you very much for your kind attention. Thank you.